Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. Thank you for joining us on this bonus day of your year. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me today from NCCAT is Ed Eng, and joining from the Pacific Northwest Cryo-EM Center, PNCC, are Kirsten Wheeler and Nancy Meyer. And from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt. PNCC has invited today's speaker, Roger Castells-Graells, who's joining us from UCLA today. And Nancy will introduce him properly after you hear a quick update from each of the 3 centers, letting you know what's new in the resources that we make available at no cost to the research community. This webinar is a regular event. We host the last Thursday of each month to highlight methods and tips and tricks that groups are using to collect and interpret the data they can obtain with the help of National Cryo-EM Center resources. So you can save the date already for our talk on March 28th. Professor Wolfgang Petty from the University of Connecticut will be giving a talk on using cryo-EM to understand dynamic interactions. I think a topic most of us are interested in. So if you want to join us for that talk, the registration link will be on the events page at cryoemcenters.org soon. And on that page, you can also find recordings of past talks and announcements for upcoming events. Back to today, please send questions for our speaker at any time using that Q&A box, not the chat, but the Q&A. And Nancy will be moderating questions at the end of the talk. So to get started, a quick update from each of the centers. And Mike, I'm going to let you go first.
Good morning/afternoon. I'm Michael Schmidt. I'm one of the co-directors of Stanford SLAC Cryo-EM Center. At Stanford and SLAC, we have 3 Krios microscopes and 1 to be installed this next month. We have two other microscopes, a Tundra and a Glacios, which will be used in our training and screening programs. We hold workshops almost every month, especially on specimen preparation and data collection and image processing. And we currently have 50 active screening projects and 10 trainees per month come through. So visit us at the Stanford SLAC Cryo-EM Center. We're glad to help you.
Thank you, Mike. Now I will turn over to Ed.
Hi, I'm Ed, here in the West Harlem Research Corridor, where NCCAT is located. We offer training, screening, and data collection access, but what I want to sort of dwell on is that this summer at the ACA, the Structural Science Society annual meeting, all the national centers are holding a workshop. So if you want to see us in Denver, sign up for workshop 3. It'll be hands-on cryo-EM sample preparation and data collection using cryo-EM merit badges. Also, what we're going to be doing this fall is we're going to be holding our 2nd annual Integrated Structural Biology Symposium. That's going to come up in October. So if you're interested in participating and speaking, let us know. If you want to find out more, please go to nccat.nysbc.org.
Thank you, Ed. And I will turn over to Kirsten.
Hi, my name is Kirsten Wheeler, and I'm the Administrative Coordinator at Pacific Northwest Cryo-EM Center. PNCC has 2 types of proposals, first of which is our screening access proposal, which allows one user to come on site for up to 5 days of sample preparation and screening with staff. This proposal is aimed at low resource institutions. The second proposal type is general access, which awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through to data processing. PNCC has 5 microscopes. We have an Arctica with K3 camera for screening and 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. 3 of our Krioses are equipped with Gaton energy filters. For sample preparation, we have 2 Vitrabots, a Leica GP2, and a Vitrajet undergoing validation before offered to users. We're also currently working on installing a Glacios 2. Installation of that has been ongoing. If interested, please go to pncc.labworks.org and apply today. Deadlines for proposals are the 1st of each month.
Thanks, Kirsten. So we have a really good talk for you today, a great talk. As you know, we're going to be hearing from Roger Castells-Graells about his current postdoctoral research in the lab of Todd Yates at UCLA. Roger received his Ph.D. in 2019 in the lab of George Lomonosov in the UK at the John Innes Center. And there he used cryo-EM to study viral maturation. And I think that prepared him well to undertake what I think you're going to find is a really creative design-based approach, employing cryo-EM to explore cell delivery methods and push the boundaries of what we're able to resolve in terms of mass using cryo-EM. We're very happy to have him presenting today. And with that, I will leave it to Roger.
Thank you so much for the kind introduction. So I will share my screen. Can you see my screen?
Yes, it looks great.
Amazing. Thank you so much. Well, again, thank you very much for the opportunity to be here today. I'm going to be talking about how in the Yates lab, we have been working on developing these protein cages to study small proteins by cryo-EM. So cryo-EM, it's an amazing tool that allows us to study proteins. And proteins, they are amazing. They have lots of different shapes and sizes. They can form like large assemblies, like for example, viruses. They can form like fibers. They can also form complexes with nucleic acids. And we can study proteins usually in vitro, but nowadays we can also study them in situ. And if we look, for example, at the cells, what we can see is that inside the cells, lots of things are going on. And if we go inside, what we may be able to see is something like this, an amazing world of proteins and other molecules working together, coordinated as nanomachines. And if we use techniques like, for example, cryo-EM, we can currently study large things, like, for example, organelles or like some complexes inside the cell. But if we want to study smaller things that you can see in this picture, it would be difficult or not possible because they are too tiny to be seen. So one of the questions is, can protein design help us study and also modify this cellular environment and to study all these amazing proteins? So the answer is yes. And one of the ways to do it is we design protein cages. And protein cages, we can imagine them as, for example, like Lego building blocks. So you can have your Lego building blocks and you can assemble them to form larger structures. The same we can do with proteins. You can take, for example, proteins and oligomeric components. We can engineer the interfaces in between them so then they can assemble into a larger structure, which is symmetric. Like, for example, a design protein cage. And then we can use these protein cages for multiple applications. Like, for example, drug delivery, gene therapy, and bioimaging. And in today's talk, I will be focusing mainly on bioimaging applications, and in particular, how we can use it in cryo-EM and for in vitro studies. So very briefly, I will talk about what is cryo-EM. So cryo-EM is a structural biology technique, and basically how it works is you have your protein sample that you have been working very hard for months to purify. So then you put this protein sample on a microscope grid, then you remove the excess of sample, and then you freeze it very quickly. You freeze it so quickly that then what happens is that the sample forms a layer of vitreous ice, a very thin layer of ice that then we can put into an electron microscope. So here we have our sample, and then an electron beam goes through the sample and generates 2D projections at the bottom where there is a camera or, like, for example, a direct electron detector. Then we can take all these hundreds and thousands of images, and then we can select the individual particles for our proteins or complexes and use multiple software tools to then obtain 3D reconstructions out of these projections. And cryo-EM is an amazing technique. And for example, as I say, it has been used to solve the structure or lots of very large complexes. But when we start going below 100 kilodaltons, we can see that it's the minority of the structures that are obtained. And if we go below 50 or 42 kilodaltons, then it becomes more difficult or nearly impossible to obtain structures. But if we look at the distribution of the sizes of proteins in prokaryotes and eukaryotes, we can see that lots of them, they're within this range that is difficult to access or inaccessible to cryo-EM. It's true that some of them, they are forming larger complexes, but some of them, as I say, we cannot study them. So here in this case, you can see an image from the cryo microscope, and here I have highlighted some of the particles that corresponds to our protein complexes. In this case, corresponds to one of these protein cages that is working as an image in a scaffold, and I'm going to talk about more this now. So one of the important things in cryo-EM is that you can see your particles. I say, if your protein is too small, you will not be able to see it, and then you will not be able to do the reconstruction. So what we want is larger things. And just to show you in a video, here we have our sample with our proteins sitting in the eyes, and then we are taking an image. So this is the 2D projection of those views. And then what we need to do is to select our particles. So they need to be large enough to be able to identify them. And then we can, for example, classify them. And what we are seeing are the different orientations of these particles on top of the grid. And then what we need to do is to find each one of these projections, how they are oriented in the three-dimensional space, to then be able to find the right orientations and then create these 3D reconstructions. So you can see how we are adding more and more particles, finding their orientation in the three-dimensional space, aligning them, and then creating this high-resolution structure. As I was saying, I'm going to tell you how we are using these protein cages to now study small proteins. So the idea is that we have a lot of small proteins that we cannot study with cryo-EM, and then we attach them to a protein cage. And then what we have is a much larger assembly that now we can identify and see in the cryo-EM, and then we just need to collect data and then get the structure for the whole complex, and then isolate the information for the small protein of interest. And now the idea is that we will have lots of new protein structures. And here you can see, for example, a protein that is too small to be seen, but now we are attaching it to this scaffold. We put it in the cryo-EM microscope, and then we collect the data, and now we can solve this protein structure of interest. So I would like to tell you a little bit about the approach that we have followed to develop this technology. So in this case, here you can see a protein cage, which is made of two components, cage subunit A in yellow and cage subunit B in orange. And we have fused to cage subunit A something called a DARPin. And DARPins, they are like antibodies. So they have these loops here that they are like fingers that they interact with your protein of interest. So what is possible to do is to generate libraries of DARPins with millions of combinations for the residues in these loops. And then using different techniques, like for example, yeast display or ribosomal display, you can then select the DARPin that has the highest affinity against your protein of interest. And then you sequence that, and then you can integrate the sequences of that DARPin into this design, and then you can make it specific for your protein of interest. And here you can see, as I say, the two components, now this binding site. So then what we do is we add our cargo protein, that in this case it has affinity against the DARPin, and now you can see the formation of the whole complex. And this tool is useful not only to solve small proteins, but as you can see, it has symmetry. In this case, it's tetrahedral symmetry. So it has 12 binding sites. So it means that for each image that you get for one of these particles, you're already getting 12 different orientations, which is very useful to avoid problems with preferred orientation. Also, there is always a site of the protein cage that is away from the air-water interface. So if you have problems with that, then it's possible when you do the data processing to break the symmetry of the complex and then treat each subunit individually in order to preserve the ones that are fine. And we have also optimized this method to get very nice distributions of particles on the grid. So we get now 400 to 500 particles per view usually, which with the T12 symmetry, then this would mean around 6,000 particles per image. So the initial designs that were developed in the lab, here you can see the protein cage, and in salmon, you can see the DARPins. Each DARPin was, let's say, on their own. So they were connected to the protein cage with a helical fusion, but the resolution that it was achievable was limited by the fact that there was flexibility in between the DARPin and the protein cage. So then I worked on a new generation of these protein cages where we oriented the DARPins, here also in salmon, in a way that they would come together and form a trimer. And then using a tool called Rosetta, I engineered the stabilizing context in between the DARPins in order to rigidify the assembly. So by creating this additional context, we reduce the ability of the DARPins to be flexible. So now we have a much stable complex, and now we can achieve a higher resolution. And this is another view of these designs. Here you can see the DARPins, how in this design, each of them is kind of on their own. And now in this new design, where we oriented the DARPins to form a trimer, plus the additional interfaces, now we can achieve high resolution, and we can achieve 3 angstroms and even sub 3 angstrom resolution for the proteins that are then interacting with these DARPins. And this is just to show you a little bit about the degrees of freedom that this DARPin has when it's attached to the protein cage with this helical fusion. Here you can see all the different motions that we can model for it. So here in this area is where we introduce this engineering approach to create new interfaces to then stabilize it. And we keep these 2 interactions that are the ones that make the protein cage as they are, but this is the place where we do the engineering. So we have started working on quite a few designs, and some of them work, and this was pretty exciting the day that we went to the electron microscope and we could see the protein cages. Here you can see these, this is negative staining images, stained with 2% uranyl acetate, and you can see these small cubes here. And here you can see this other image, and for example, you can see 2-fold views. These are the protein cages that we had designed. And when we saw this, it was like, this is like so cool. So in this particular case, the design that I made was with a DARPin that would bind to GFP as a test. So GFP is the green fluorescent protein. It has 26 kilodaltons. So if I put, for example, it through one of these filters that has a cutoff of 100 kilodaltons, what you can see is that the GFP goes through it. But now if we mix the GFP with this cage, with these binding sites, now they form this 600 kilodalton complex that you can see it is retained here on top of the filter. So then we froze cryo-EM grids, and you can see that we get a very nice distribution of particles and different views. So it was like, okay, we are ready now to collect high resolution data. So We went to the Titan Cryos, which is a 300 kilo electron volts, a high performance transmission electron microscope. And we collected data for these designs. And you can see here how we have lots of particles. And in this case, we were getting about 400 particles per image. But as I say, as it has like tetrahedral symmetry, then it goes into the thousands. And we pick the particles, and now here you can see when we do the classification of the particles, and you can see now the 2D projections, which we call 2D classes. What you can see is that for the protein cage, we can already see secondary structural features that, like for example, these tubes here that would correspond to the helices. And the exciting thing is that you could already even see features and the density for the GFP, which is this 26 kiloton protein attached on top of the scaffold. So now I'm going to show you a 3D reconstruction of one of these binding sites. So here you can see a focus view on one of those DARPins, and here on one of the GFPs bound to it. As I say, this is pretty cool that now we can see a protein like GFP that is 26 kilodalton with cryo-EM, and we can see a nice secondary structural features. And if now we, for example, cut through this GFP, through this barrel, and we go inside it, what you can see is very nice density, for example, for the chromophore, and you can also see density for the side chains around it. So this was pretty exciting. And here, this is the composite map for this structure. And in gray and blue, there are the two components that make the protein cage. Then in salmon, we have the DARPins, and in green, we have the GFPs. And this assembly is tetrahedral, so we have 12 copies of the GFPs, and as you can see, they are in different orientations. So this is pretty cool, and we all like GFP because it's green, it's fluorescent, it's easy to work with. But the question is, could we use this technology for other proteins, for other targets of interest? So could we potentially use these imaging scaffolds as a modular tool to solve the structure of any small protein of interest? The answer is yes. So as I was talking, DARPins, they are made of these loops. And basically using these libraries and using methods like yeast display or ribosomal display, you can select DARPins against your protein of interest as long as they can form an interface. So, and then when you have found the DARPin that has the highest affinity against your protein of interest, you just sequence it, and then we can integrate the sequence in these loops into our design. So now I'm going to show you an example for when I took the sequence for a DARPin that would bind to KRAS. And KRAS is a 19 kilodalton oncogenic protein target that is of great interest for the pharmaceutical industry because KRAS is involved in 25% of cancers. So there are a lot of efforts to develop new drugs and treatments. And we have developed this work in collaboration with AstraZeneca. But as you can see, as KRAS is 19 kilodaltons, it's too small to be seen on its own with cryo-EM, and it's a protein that you can crystallize. But sometimes, for example, if you want to screen for ligands, it's possible that some of them may affect your ability to crystallize it. So then using alternative methods like cryo-EM is when it becomes useful. So I took the sequences for these DARPins that would bind KRAS, we integrated them into these protein cages. And now here you can see we will mix the protein cage with KRAS. And here, this is a size exclusion chromatography plot. What we can see is that first we get a peak that could correspond to the imaging scaffold plus the bound KRAS. And then later on, we get a second peak, which corresponds to unbound KRAS. And we have unbound KRAS because we usually add it in excess to ensure the maximum occupancy on top of the protein cage. And here in this image on the right, what you can see is a SDS PGL of the fractions from these peaks. So the first peak, you can see that we get 1 and 2 bands that correspond to the 2 components of the imaging scaffold. And then you have another band which corresponds to the bound KRAS protein. And later on, in the second peak, then you get the excess and bound KRAS protein. So we froze grids and we collected data. And as you can see, we get a very nice distribution of the protein cages on the grid. Again, in this case, we're getting about 500 particles. They have T-symmetry, so it means that we would get about 6,000 individual particles per image. And here on the right, you can see the 2D projections. And again, you can see secondary structural features for the protein cage, like the helices, that could be the rods. And you can already even see the density for the KRAS on top of it, which in this case, I say it's 19 kilodaltons. So we did the 3D reconstructions of this. And here you can see the composite map. Again, you have in gray and in blue the 2 components of the protein cage. In salmon, the DARPin, which is the binding site, and then in yellow, the KRAS. So, and here now you can see the 12 different views of this KRAS on top of the protein cage. And when we got this, actually was like, wow, this is like so cool. And we can see now great detail for KRAS. We can see ligands bound to it. Like, for example, in this case, we could find the density for the GDP, and also, for example, for magnesium. You can see like side chains. And this was really good. And then the question was like, okay, now we can solve other proteins, like for example, KRAS, which is 19 kilodaltons. But could we now use this technology to screen for, like, for example, drugs that are, or ligands that are interacting with these small proteins? So we collaborated with the University of British Columbia and the Gandiva Therapeutics because they were interested in studying proteins with cryo-EM and to develop and to study, for example, how drugs interact with them. And they were like, you want, you can come here to Vancouver. We will give you microscope time and we can test how this works. And I was like, sure, let's do it. So I traveled to Vancouver and we did the following experiments, and I'm going to show you some of the results that we got. So in this case, we tested KRAS and a mutant called G12C, and we tested how to get the structure bound to a drug called AMG 510. AMG 510 has been FDA approved, and it's a drug from Amgen used for lung cancer. So we could see and solve the structure of now KRAS bound to this drug. And if we compare the cryo-EM structure that we got in green with a previously known X-ray structure in red, we can see that there are small conformational changes, for example, in the drag conformation and also in this loop here. And for example, if we look at the crystal structure, what we can see is that there are some crystal contacts around this loop here. So it's possible that the crystal has constrained a little bit the conformation of the protein around this area. But what I think this data is showing us is also the power of using and combining multiple techniques. So cryo-EM and X-ray in this case, they are showing us 2 possible conformations of this protein and this drug. And then this gives us like a broader view. So yeah, it can be really useful to use multiple techniques to study your samples. And here you can see the structure for KRAS when it's not bound to the drug, and now here when it's bound to it. And you can see there is a conformational change. And what the drug does is it locks the KRAS in this conformation and in the GDP form. And here you can see now the morphing between the 2 structures. And if you are interested to learn more about this, we have published this work And now we have also like a patent. So if you want to access this technology, you can also contact with the patent office at UCLA. So the next question is, can we use these protein cages inside cells? So could you use these design protein cages, for example, for drug delivery, gene therapy, and in our case, for example, in bioimaging? So we collaborated with the lab of Professor Hilary Kohler here at UCLA to do a few experiments, and I'm going to show you now a couple of the results that we got. So this work has been led by Morgan G, who is an undergraduate in our lab, in the EH lab, and by Kaiser Atai, who is a graduate student in the Color lab. So the idea was to deliver these protein cages, and we tested to do it in HEC293 cells, which are mammalian cells. And the idea is we would prepare these protein cages. We label them with Alexa Flor 647, which is for far red. We tried multiple ways to bring them inside the cells. And in our case, the one that was working the best was using a liposuction. So we did liposuction to then transfect the protein cages inside the cells. Now I'm going to show you an example where we had a cell that was expressing GFP on the cytoplasm, and then we added protein cages that had DARPins against GFP. So here, this is the control where you can see the GFP in the cytoplasm of the cells. And now we add the protein cages inside the cells. And what you can see is that this green puncta start to form. And if we look at the Alexa Flor 647 channel, which corresponds to the labeled protein cage, we can also see that there is this puncta forming, and we can see that there is co-localization between the two. So we see co-localization within the protein cages and now the cytoplasmic GFP that is interacting with them. Yeah, here you can see one of the examples. So, okay, that's great. We can now bring the protein cages that are fluorescently labeled, bring them inside the cells, and now interact with a cytoplasmic protein, like for example, in this case, overexpressed GFP. But now the question is like, can we make these cages go to a specific place in the cell? Can they target, for example, an organelle like mitochondria? So for this experiment, we used MFN1, which is a membrane protein from the mitochondria, which was fused to GFP. And this GFP was exposed on the cytoplasmic side. So it was accessible. And then we did the same experiment. We prepared these protein cages, fluorescently labeled with Alexa Flor 647. We added them inside the cells. And what we can see is that then this puncta and then if we look for the GFP, we also see that MFN1 is localizing in the mitochondria. It forms this puncta, and then we see the co-localization between the two signals. So this is very exciting for us because now we can bring protein cages inside the cells, and then they can target, for example, a protein that is found in an organelle. And in this case, we have been using GFP, but now you can just imagine engineering the DARPins on these protein cages against any protein of your interest, and then having them go to that specific protein. So as you have seen, protein cages, they can be very powerful. And in this case, the one that we are using has been designed. So when we are developing multiple applications, sometimes we need protein cages with different shapes and sizes. And for example, the ones that we used, they work in particular conditions, but you may say, oh, I want to work with a protein that has a different pH or that is stable in different buffers. So could we have more protein cages? Because at the moment there are some, but the amount that is available is limited. So how else could we expand this protein space? No? How else could we create more protein cages? So I would like to show you a little bit about the power of artificial intelligence in protein design. So the idea of the following project was to design new symmetric assemblies with machine learning algorithms in order to expand the available protein cages. And the idea is that now you will have like a large tool set of protein cages. So now you will be able to select those that fit better the needs of your project or the questions that you're trying to answer. And for example, to develop new imaging scaffolds with different properties. And we did this work together with Kyle Medor, who is a graduate student in the Yates lab. So basically, the idea was to use multiple tools, and one of them is called Protein MPNN. And this is an amazing tool developed by the lab of Professor David Baker at the University of Washington, which allows you to design new sequences. So I will explain you a little bit how this works. So basically, you can imagine a protein monomer, and then these protein monomers, they can form, for example, oligomers like trimers. So what we did was to dock two different trimers in a way in the three-dimensional space that then they will assemble into a symmetric assembly. Like, for example, this is a tetrahedral assembly. And then we would put these structures and provide them to protein MPNN and tell it, generate us a new sequence that it's going to be able to fold in the same way as the subunit. At the same time, it's going to make contacts with the two neighboring subunits in the trimer. So it's still going to form a trimer. But at the same time, it's now going to form contacts with the subunits from the neighboring trimer, which is different. And the same for the subunits in the other trimer, in the purple one. It's going to redesign it to have the same fold and then form interactions to still form a trimer, but at the same time now form interactions with the subunits from the green trimer at this particular angle to then be able to form this three-dimensional structure. So initially, I thought that this would be like magic to be able to do this, but it works, and I'm going to show you a few examples. So to do this work, we had to look and screen to lots of different designs, and we have been collecting data here at UCLA, but for us, it became instrumental also to be able to have access to the NIH cryo facilities. And I would like to thank Patrick Michel from S2C2 at Stanford, and also to Omar Dabulko at the PNCC in Portland for their amazing help and work to collect some of the data that I'm going to present you now. So for these two-component protein cages that we designed with these machine learning algorithms, we were able to solve 7 cryo-EM structures. And here you can see a few of these structures. You can see they are made of two components. One trimer is in green, the other one is in purple. And you can see that they have different shapes and slightly different sizes. And they are made from original different components. And so they have all like different protein sequences. And here in the bottom, you can see in gray is our initial models, and in color is the cryo-EM structure. So it was quite amazing that the cryo-EM structure matches pretty well already the initial models that we got. So now I would like to show you a few of these structures, and I will start with TT3ML23. This data set, for example, we collected at S2C2 in Stanford, and it was like pretty cool because you can see here we would get a very nice distribution of the protein cages, and here you can see that there are here already some secondary structure features, like for example, some helices. And for this one, we managed to get to 2 angstroms, or about 2 angstroms. And this was kind of an amazing day, the day that it appeared like 2 angstroms. It was like, wow, this is like so cool. The first thing that we did is like, can we see some features in the side chains? So the answer is yes. So here, it was quite exciting to be able to see, for example, now holes in some of the side chains in the amino acids. It was like, wow, this is like so cool. So yeah, so now we can design these new protein cages synthetically, and then we can solve their structure with cryo-EM, and they are so stable that we can now get, in this case, to Armstrong's resolution. And here you can see two views through the threefold of this assembly. Here is one of the trimers in green. Here is the other trimer in orange. And here you can see it. For example, this protein cage has here a pore. So you could imagine that you could use it, for example, to also like as an reactor, or to load it with, for example, like a drug, to use it for like delivery. So this could be used for multiple applications. And here you can see it spinning around. And another thing that is great about cryo-EM is that it allows you to study heterogeneous samples. So here you can see a lower resolution reconstruction of some intermediates in the assembly process. So in this case, this protein cage is missing one of the trimers that this could correspond to this one. So yeah, so you can see that for us, this is important because this way we can also learn about how these interfaces are working, which are the ones that are stronger, how these are assembling, and this gives us valuable information that then we can use to iterate and create new designs and fine-tune them for the specific properties that we want to use them for. Here, I will show you like another design. In this case, it's called, well, we call it design F2. Here you can also see a very nice distribution of the particles on the grids. Some of this data that I'm showing now is also collected at the PNCC in Portland. And you can see here also that we can get very nice secondary structure features already in the 2D classifications. We could get also a very nice 3D reconstruction. And in this case, here you can see two different views. In this case, it has a slightly different trimer here. In this case, you can see how this one has this structure here that's more closed, this pore. And another interesting thing that we use these structures for is to understand how the interfaces are being formed, and if the new amino acids that we are engineering to form these new interfaces, how they are working, and if we can, for example, introduce mutations to make more contacts and then make more stable connections, or if we can engineer them to make them even weaker, so to have more dynamic assemblies. So yeah, so this structural information for us is very valuable to then be able to fine-tune these assemblies. And here I will show you another one of the designs. In this case, this design, one of the views, it looks like a triangle or like a ninja star. And in the other one, it looks like another view. So if you look at different orientations, it looks like a cube with like spikes on the sides. And in this case, the fold that we used for one of the components was a protein that was already originally synthetically designed. So we redesigned already a protein, but in this case, this protein was just forming a trimer, and now it's forming part of a larger complex, which is also a tetrahedral assembly. And here you can see how this one looks like. It's also very porous, but still we can get to sub 3 Armstrongs for these structures. So yeah, so here I have shown you a few examples of some of these designs. If you want to read more about this work, we posted this in bioRxiv, and now it has also been accepted in structures, so you will find it posted soon. And if you're interested in this technology, we also have a patent for it. So I hope that I have shown you a little bit about the power of these symmetric protein architectures, and that these cages, they can be engineered in rational ways to function as high-resolution imaging scaffolds. And we can see that the design of these additional interfaces in between the DARPins then rigidifies the assembly, allowing us to reach this high resolution. We can engineer then the loops in these DARPins in order to change their affinity against the protein of interest. So this is a modular system. And we have developed this for in vitro studies, but we are also working for in situ studies. So to conclude, I would like to acknowledge lots of people that have been helping to make possible this work. So I would like to thank Professor Todd Yates here at UCLA for all the amazing opportunities and all the support and the chance to work in these very cool projects. And then I would also like to thank everyone in the Yates Lab. In particular, I would like to mention Kyle, Morgan, Nika, and Justin, with who I have been working more closely for the development of some of these projects. I would also like to thank Professor Hilary Kohler and Kaiser Atai for the amazing collaboration with the cellular studies. I would also like to thank everyone in the core facilities, microscope facilities that makes possible to get this data, to keep running the microscopes. So I would like to thank Dulio, Michael, Alex, Genesis, Mark, Tom, Peng, and David from UCLA. And I would like to thank Patrick from S2C2, and then Omar from PNCC. I would like to thank also to Jack, George, Stomu, and Tatiana, who are collaborators and mentors. And then I would also like to thank to our collaborators at AstraZeneca and Gandiva Therapeutics. I would also like to thank our sources of funding, the NIH and the Department of Energy, and then also the NIH cryo-EM facilities, again, for this amazing resource. I recommend everyone in the audience to look into it and to apply for it if you have a project that you think that could be interesting to look with cryo-EM, because for us it has been a very useful tool to be able to do our projects. And then if you want, you can also follow us on social networks. And yeah, I would be happy to take any questions.
I think you're missing, well, in Zoom, we're missing the loud round of applause. That was absolutely amazing. You probably couldn't see me. I was like shoving my face against the screen, looking at all of your gorgeous, gorgeous 2Ds.
Thank you.
Thank you so much.
We do have a number of questions from the audience for you. First up, do you worry about the DARPin target interaction resulting in conformational changes of the target protein?
Yeah, this is a great question. So for the structures that we have solved so far, we have compared the cryo structure that we obtained with the protein bound to DARPin with proteins that were not bound to a DARPin, like for example, that they were crystallized. And we have seen that there are not conformational changes induced by the DARPin. In fact, DARPins, they are very sensible to conformational changes. For example, the DARPin that binds the KRAS GDP form, if you want to bind to the GTP form, then you need to use like a different one because the conformational change that it induces has an effect. So in fact, the examples that we have tested so far, we have seen that is the DARPin the one that is more sensitive to like conformational change of the protein itself. So we have not seen any induced conformational change.
Thank you. We have a great question. Please tell us about the difficulties that you faced and how long it took to make the DARPin strategy work since your first steps. And did you start the several different constructs simultaneously?
Yes, this is like a great question. So I would say, so we started this project a few years ago, and it took us, yeah, over 2 years to complete. So, and yeah, at the start, we were using protein design tools at the time, like for example, Rosetta. Now there are new tools, like for example, protein PNN and lots of others, which are speeding up things. So in fact, protein design is becoming a very, very exciting field to work on. So to answer the question, so we started making a few thousand in silico designs of these oriented DARPins, and then we applied multiple filters, and then we started to narrow down, and then we went to like a few hundred, and then we inspected them visually, and then in the end we selected a few tens of them that we then expressed. And then for these ones that we expressed, some of them, they were insoluble. Some others, they will not assemble properly. So in the end, we ended up with 5 designs that would form protein cages and that would have like the right arrangement. And then we look for the design that when we would do cryo-EM on it, would have the highest stability and that would yield the highest resolution. And then for the DARPins, you can get them from the sequences from publications. But for example, if you want to raise them yourself, it can take from weeks in some labs to, in our case, it takes us a few months to raise a new DARPin, and then you just need to then integrate the sequence of that new DARPin into the scaffold. But you just need to change the loops. You don't need to redesign the whole thing.
Thank you. Another question we have, is the DARPin binding to only one side of the protein? Have you tried not too small proteins that cannot be crystallized because of flexibility? So have you tried larger proteins that are only not viable via EM because of flexibility? This person suggests that they work with 130 KD protein that could have like 2 binding modules. If the cage would immobilize those modules together or separately, is it feasible that they could solve that structure of something that large?
That's also a great question. So in our case, the DARPins usually they are very specific for where they bind. But when you select them, you can get multiple sequences of the DARPins that bind to multiple areas of your protein. So, for example, if there is an area that you are interested and that maybe you want to see how a drug interacts with that area, then you can select a DARPin that binds to a different area, so that area that is free. So you can screen your protein by selecting multiple DARPins. Then for the size, the maximum that we are being able to do with this particular design that I presented today is around 45 kilodaltons because of the way that the DARPins are oriented. But we are working on new designs where the DARPins are oriented in different ways, where then you can solve larger assemblies. And the idea is that it could be useful, for example, like in the case that was mentioned, or for example, in cases where you may have problems with like preferred orientation. So then this would be our robot interface. The assembly will already give you like all these different views. So
Give you
for the
variety,
particular
yeah.
design with this 130 kilodalton protein, it will not work. But stay tuned.
Yeah.
We may have one soon. But you can also reach to us if you want to further talk about this.
And I think our next question may have been answered by that. Can we use this technique for proteins that oligomerize or form heterodimers? Do you think the DARPins will have steric clashes with the higher oligomers? And I'm guessing that relates to the steric clash of having larger proteins.
Yes, in our case, by the way that they are oriented, yeah, if you put something too big, then there is going to be like clashes.
And how do you envision to connect new small protein in your AI-generated protein cages? Do you have them tested with other small proteins yet?
So
maybe some of the new AI-generated protein cages, how would you have them interact with new small proteins?
Okay, yes, this is also a great question. So basically what we built with the protein cages is what we would say like the scaffold. So now as these protein cages, they have different termini, different orientations at the ends, then you can engineer in top of them like these binding sites. And in this case, you could use, for example, like DARPins, like if you have like helices, but you could also imagine other binding proteins. And then when those are engineered, then you could make them bind to your small protein of interest. So the protein cage that we designed so far is just a scaffold, and then your imagination is the limit. So then you need to think, oh, what I want to use it for. And if you wanted to load a drug, you can maybe add specific binding site or specific charging site. But if you wanted to now display another small protein, then you could engineer now a protein that is able to bind to things, like for example, a DARPin, like we have done in this example.
Great, thank you. Okay. We've got a few more questions that just came in. Complementing the talk, can you explain the basic workflow to get to the final structure you showed? Like the sequence of jobs in, for example, CryoSpark. And I think maybe they're asking about the basic steps of the focused refinement to see the appendage proteins separate from the cage.
Yes. Yeah. No, this is also a question. So basically, we would follow a standard data processing flow initially, like all the steps, motion correction, CTF, all this stuff, just like your particles. Then we would do a reconstruction using the tetrahedral symmetry, which will usually yield to a very high resolution already for the core of the assembly. And then to do the focus part, then what we do are multiple steps. We can, for example, apply a mask that encompasses one of the subunits, and then we can break the symmetry. So and then treat each one of the 12 binding sites individually. And then for here, we then do steps like, for example, classification, 3D classification without alignments, in order, in case that there is like a binding site that is not occupied, or that maybe disrupted by interacting with air-water interface. And then we clean this data set to keep those ones that are the best. And then here, then we apply also some then like fine local refinements to try to then also resolve for like maybe so much like remaining flexibility. But yeah, basically it would be this. We do first the whole assembly, and then we break the symmetry, and then we treat each one of the components individually, but already the orientations that we get from the whole assembly, they are already quite accurate. And we can already see the structure for the components on top. It's tend to get to the higher resolution when we need to do these local refinements, extra steps of classification.
Sure.
And
Thank
actually,
you.
if I can ask a quick follow-up, do you generally
Please.
find the same workflow works for each target, or do you sort of need a custom solution each time once you drop symmetry?
I have to say that we used a slightly different approaches, but it was also because we collected the data different times, and new tools were appearing. So there were softwares in the updates in the software too. But for example, for the KRAS, we solved 4 or 5 different structures for different mutants and so. So once we had one specific protocol, then we could apply it quite quickly to the others, which is very nice because then it would shorten our processing time to just like a few days or weeks. So yeah, so you can adapt the protocols, but sometimes it's just that the new tools or ideas go so fast that then you just then say, okay, I already got this structure. I don't want to reprocess it. I'm going to use this new approach for this new one. But yeah, you can, lots of things that you learn for one of them, you can then apply it for other structures that you want to solve.
And I'm going
I
to
think
interrupt Nancy for just a second. We have a
Yeah.
number of questions left, but we're coming up on the hour. So for those of us who have to go, thank you for joining us. We're going to stick around another couple minutes and get to a couple more of these questions, and we'll keep recording. So if we didn't get to your question yet, come find the recording, and we will stick around and get to a few more of these questions.
Yeah,
All right,
thanks.
go ahead, Nancy.
I think the questioner might be interested in knowing which software package you were using for some of these.
For the design or the data?
Oh, I'm sorry, for the focused refinement.
In this case, we recently have been using more CryoSpark.
Okay.
I would say that we have been iterating between Relion and CryoSpark and so, but recently, well, as I say, it depends a lot on the specific steps sometimes that you want to do. But recently we have been using more of the CryoSpark local refinement tools. But I think that both tools are really good. And as I say, I would say it depends on which step you want to do and also the resources that you have available.
Thanks. Our next question is, could you please tell us more about the method, how to quickly find an appropriate DARPin for any given protein? Do you have a library to screen for each, or do you design specific DARPins for each new protein? And what's the advantage for the cage target protein over antibody target protein method?
Yeah, these are all really good questions. Thank you so
Yes,
much.
so we'll take maybe the first one about the
Yeah.
finding the appropriate DARPin.
Yes, so the way that we do it is experimentally at the moment. So basically you build first your DARPin library with like all the possible combinations in these amino acids in the loops. And then you can use, for example, yeast display. So then what you do is you take your target protein and you put it against the library with use display. And then you do multiple rounds of selection. And in each one of these rounds of selection, you keep those that have the highest or stronger affinity against your target. Then you do multiple of them, and then in the end, you sequence your best targets, and then you express the DARPin that encodes for those particular ones, and then you test the affinity against your cargo. And then you check, for example, like the KD. And then for when we have checked that, then we take the sequence for the specific DARPin, and then we integrate it into the imaging scaffold. And there are multiple ways to do it. Some people also use like ribosomal display. Yeah, there are multiple approaches. The advantage of doing it experimentally is that you don't need to know anything about the structure of the protein. So you may not know how it looks like, or yeah, where it needs to bind. As you are just screening for it, then you just are able to select it and then to study it.
And the second half of that question is, what's the advantage for the cage target protein versus antibody target protein?
Oh, yes, yes. So in our case, the cage is usually like a much larger assembly. So, and also, as I said, this has symmetry. So in our case, the symmetry helps a lot to get all these different orientations, also to avoid air-water interface. And in our case, we use DARPins on top of the cage too, instead of, for example, like nanobodies, because we could fuse the DARPin terminal helix to a terminal helix from the protein cage, which then already gives us like a better starting point if we want to then reduce the flexibility. While for some of these antibodies or other proteins, the linker between the two would be much more flexible. So we would be starting from a more like floppier point.
Can you use the cage DARPin GFP to look at the GFP fusions, or are would the fusion be too flexible? Rather than making new DARPins each time, can you just bind stuff as a GFP fusion?
This is also a real good question, and we are also wondering it at the moment. Okay. I think that the problem is usually the fusions between the GFP and the target protein would be too flexible. The thing is, the idea is that to use it, for example, like as a marker too, so you could see where is localized your specific protein, and maybe if it's like a complex that is not as easy to identify, but if you have a better idea of where it is, it can help. That could help. But if it's like a small protein that is just fused like to the GFP, in that case, yeah, I think it would be like impossible to solve it, at least with standard methods. But in that case, it's more like to see at high resolution where you find your protein, and then yeah. But yeah, this is a really good question, and it's something that we are also currently exploring.
We do have more questions. Do you have any suggestion for improving the flexible region fitting in the EM volume? Improving the flexible region fitting, that's how they're saying it.
Well, if it's to solve flexible things, what I would say, we are usually using like, we use multiple masks, and then we do, as I say, all these specifications and refinements around our protein of interest. Sometimes you can also do like multiple steps, like boxing it out and signal subtraction. There are like multiple methods that have been developed to try to resolve flexibility. But I say usually flexibility, if you want to get high resolution, is not good for cryo-EM. It can be interesting to learn more about the dynamics of your system, but if what you're aiming is at resolution, what you want is to have it more rigid as possible.
Hi, this is Mike Schmid. As a shameless plug for other methods of looking at flexibility, you might try the Gaussian mixture model that was developed by Muyan Chen, who's now in our lab, that deals specifically with looking at the flexibility and including including the flexible part and the stable part, rather than solid and rigid masks between them. It seems to work very well. And again, shameless plug, but it's something that that you should look into.
Yeah, absolutely, absolutely, absolutely. Yeah, yeah. And yeah, absolutely. I think when we saw that method coming out, we were very excited. I say it's something that we are also looking into trying. And yeah, I think, as I say, you see, like another method that came out quite recently. There are continually like new tools coming out. But yeah, so thank you so much, Michael, for mentioning it.
We did have a question about how feasible it is to use elongated proteins, and I think that related to the mass of the target protein. So if that person wanted to re-ask the question, please do. We have a question: where did you find your DARPin raising protocol? Is that something people can maybe Google? Do you have a
Yes.
specific resource?
Yes, so DARPins, they have been developed by Andreas Pluchtung at the University of Zurich. So first one to check would be to check all his work. And then the DARPins here at UCLA, they have been developed by our protein core facility. And it has been led by Mark Arving. And that's a great question because he just published a protocol in Current Protocols last month.
Okay.
So if you check for Mark Arving, UCLA, DARPins, and so there is like now a protocol also published on it. So yeah, you can find it. But yeah, if you can look at also at the work that has been done in the last few years by other labs. And yeah, if you check for Andreas Puchtung, you will find lots about DARPins. And as I say, it depends on the lab. They use different approaches, and some people use the ribosomal display, other people use yeast display. Yeah, depends.
Thanks, that's a great resource. Also, another complimenting on the exciting work, do you think a modified version of the system could work for membrane proteins?
Yes, the answer is yes. And it's something that we are looking into also at the moment. Yeah.
And if you have to redesign new scaffolds to optimize binding to the target, would it be useful to fuse your target to one of the subunits to make sure that all of the sites are occupied and you might have less heterogeneity?
This is a good question. We have not done it. I would say maybe in the case if you had already some clue of the structure of your protein, if you know, because if you don't know, for example, where are the termini, if it's like exposed or accessible, I think that would be more difficult. But if you have like some idea of where it is, maybe yeah, you could try to fuse it if it didn't alter it. I think it could be feasible to do it if you have some preliminary information, and I think it could be cool to try it and see the differences. Yeah, I think that it could be possible to try, but you may need to know some preliminary information to be able to make the right length of the linker, and yeah, and to make sure that then it doesn't clash, and yeah, but yeah.
Yeah, maybe a little more troubleshooting.
Yeah, yeah, yeah.
The DARPin, you can go in kind of blind, right?
Yeah, exactly. The idea of using this approach was to make it like modular and quick to work with, so that when you would have the DARPin sequence, you would just need to swap it and don't need to redesign it. Because also for when designing things, my advice is usually keep it simple. So the least modifications you can make into a protein usually, at least in our designs, it goes the better, because every time that you introduce a mutation or a change, there is a chance for trouble, let's say, for your protein to not be happy, aggregate, to do something that you don't want to. But yeah, I think it would be something worth trying to.
Okay, we have two more questions for you.
Sure.
Did you perform the 3D classification without alignments in CryoSpark? And if you did, did you use any non-default parameters that you can think of? Did you have to customize the job very much for the 3D classification?
Yes, we used well, we used 3D classification without alignments.
Yeah,
For the parameters, it's
we're
really,
really pushing you here.
yeah. It's because it has been a while and we tried lots of things. So, um, yeah, I cannot think out any specific one. I just say that we tried like multiple classes. One of the things was the masking and yeah, yeah. I think we just, as I say, we started with the default parameters and then we tried multiple things. I could go back into my notes, so if this person wants to contact me, I can then find the exact numbers. But we used pretty much usually default parameters, and then a lot of things would be influenced, for example, of like the size of the mask, and also already how good was your starting data set. It would be much easier to clean it, and so yeah.
I think you're going to get somebody contacting you. So thank you for that information.
Yeah.
And our last question, how do you decide where you want the cage proteins to bind to the target? Is it part of the design of the cage where you'll have termini with the
Yeah,
DARPin?
yeah, that's also a good question. So in this case, for the cage that we selected, we looked at lots of different protein cages. The one that we selected was the one that had the terminal helices, and that those terminal helices, they were coming at this particular orientation. In this case, we tried fusing, for example, the DARPin in multiple ways. So if you go along the helix, it will start rotating. So then find the right distance between the DARPin and the case surface, but then that these DARPins, and they will be able to form in this case like the trimers. So we had to look, at different cages and look at the terminal helices. So this is why we are, and other labs also are building lots of these new symmetric assemblies in order to make a much broader library. So then when you have a specific problem, you can say, okay, I want a protein cage that have this terminal helix pointing in this specific direction and or in this other way. So then you can make tools that are more fine-tuned. Because in our case, we had to explore all the available ones, and we chose the one that was the best available one at the time. But maybe it's possible to even make better ones now when you have more possibilities.
So you have very specific properties in mind when you're designing the new ones, or you're trying to create the new ones.
Well, properties are, for example,
In terms of
well,
how the DARPins are arranged or spaced.
yeah, in our case, when we're making the design protein cages, we're just trying to make a set of different ones that could be used for different applications. So we were thinking more it like broadly, but for example, those ones or from ones that are from other labs too, if I said, okay, I want to fuse on top of them a protein that has a terminal helix too, and I want to use the advantage of having a helical fusion to maybe have already a better orientation and less flexibility, then I would say, okay, so then what I would do is I would look through all the available symmetric structures and look for those that have accessible terminal helices. Or sometimes maybe the helix is there, but it's not accessible, but then you can just do like an extension and build synthetic helix and then test at each position in the amino acids in the helix and go up and see which is then like the best orientation. And then you can use all these protein design tools and tell it, okay, now using this particular like backbone, redesign me the residues. So now they will form like this helix and maybe it will have some specific property. So you can always also add bits and pieces to it and then to adapt it to your thing and then tell it the program to then redesign you the residues to still have the same fold and property, but to, yeah.
So all in silico before you express anything.
Yes. In our case, we do everything in silico, and yeah.
Okay, well that finishes up our questions. Thank you for sticking with us. We have a lot of interest in this.
Yeah, no, thank you so much for the opportunity to be here today, and thank you everyone for the amazing questions. I really enjoyed a lot being here today.